So thank you very much, and thank you, Steve, and the organizers for inviting me to this session. This is my first time at VISB, and I have enjoyed what I've seen so far. It's very, very motivating and lots of terrific ideas for the visualization world that we live in, the world that I live in, which as a trained physiologist, electrophysiologist, living in a world of computer geeks. I live inside the Ski Institute, Scientific Computing and Imaging, and all around me are image specialists and computing specialists, and I try and provoke them with our needs in the world of physiology and benefit from their insights. So we're surrounded by wonderful images in Utah, some of them artificial, some of them very real, and we like to think that some of that real beauty feeds into our sensitivity to visualization and how we use computing and visualization in our science. Now, this talk is going to tie into a lot of things you've heard today already, and when, with prescience, I could have said it was inspired by the talk we saw this morning, the wonderful plenary address where we looked at relationships and the whole nature of interactions between people, because I'm going to float the theory out there that visualization is a lot like relationships. There's a lot of similarity between visualization and how we do it and the nature of relationships between people. And I'm going to break that down into 6 different points, and each one of them I'm going to try and prove to you or show you evidence to suggest that these are relevant aspects of visualization, and you'll have to decide for yourself whether these are relevant aspects in relationship. And the first one of these is that size matters, no matter what we like to think. And I'll show you how that matters in visualization. You can already guess that that's going to happen, and certainly we can have long, great discussions about how it matters in relationships. Portability is a very cool thing. You know, years ago, of course, there was the great song, Love the One You're With, and we love to have portability in our visualization, too. We love to have our visualizations with us to work together in our world. Keeping perspective is crucial in relationships. We all know that. I'm going to show you that it's incredibly important in visualization. Life moves quickly. We have to keep up with it. We have to hold on to it. Life is interactive, and visualization has to be interactive. To go with that, mistakes happen in relationships and in our data. We heard that just in some of the questioning in the previous talk. We have to have a way to understand error and how it feeds into visualization. And finally, new is always better. This is sometimes true about relationships, and it's certainly true about visualization, as I'll show you. Now, I also was inspired by the talk this morning to see if there's any background or any evidence out there on the internet to support my thesis, and so I actually tried to use the visualization tool we heard about this morning to see if there was a link between visualization and relationships, and I was disappointed to see that there was actually very little based at least on this internet search. However, I was reassured because I changed the search then to say, what's the relationship between visualization and biology? And I found that there are also no relationships. And VISB doesn't show up there yet. Yes, that maybe it should be our goal. A year from now, we should see links between these worlds and between these words. And so as I said, I work in a place that's all about computing and the use of computing in science. It's the Scientific Computing and Imaging Institute. And visualization is one of the 3 major pillars of our institute. And so we have a lot of fantastic visualization specialists around and a lot of great research. And the IEEE Visualization Conference is probably the biggest one in the institute. And so I'm going to build on that basis. And just to show you also that visualization plays a big role in the research I'm directly involved with, here's just one example. This is a center funded by the NIH that is driven by integrative biomedical computing, and our goal is to provide software tools for scientists in a number of different disciplines. And these driving biological projects are supposed to motivate us to find good computational tools that scientists will actually use. So we really do talk to scientists and we do talk to physicians and try and make our tools useful. Those tools are based on image-based modeling, simulation, and estimation, but visualization is the glue that ties them all together. And this is really how we see visualization in our world of computing. It's ubiquitous, it's there at every step of the process. You've seen wonderful examples of that today. It's there when we look at our raw data, it's there when we interpret those data, it's there when we look at the analysis we perform on those data. And so this is really the overwhelming message here. This is a vision that we have at SKI about how we want to tie image acquisition together with image processing, geometry processing, modeling, simulation, and visualization, and to drive the results back into the lab or the clinical setting. We had to find some place in this diagram to put visualization, so it ended up down here in the lower left corner, but it really is a part of every one of those steps. Again, you've seen examples of that, you'll see more examples of that. My particular area of application is in visualization in the world of electrophysiology. And we use visualization at all levels. You in the room will maybe better than I do understand this diagram. It's of a potassium channel and the structure of the proteins that make up that channel. These are cardiac myocytes held together. These are models of cardiac myocytes. This is a whole heart and the voltage distribution on the surface of that whole heart. And these are animations of some data recorded directly from the surface of animal hearts, examining the spread, the dynamic process of electrical activation and spread of activation in this world of cardiac electrophysiology that I live in. Now, as I say, the visualization in our world is everywhere. It's in your world, it's in my world. These are some of the images that you can pull down off our website at the Ski Institute. And this visualization is all around us, and it's applied to all the sorts of different domains. This is obviously an embryo, high-resolution CT scan, wonderfully presented here in a visualization. Here's something very esoteric. This is the surface current flow on the surface of the heart, recorded during an instant of very complex spread of patterns where red are electrical sources and greens are electrical sinks. So we move within this broad spectrum of applications within the Ski Institute. We even visualize teeth, for example, and we have tools we've developed for a lot of these particular applications. And so the visualization is all around us. We use it ubiquitously, and I could spend most of my talk just showing you image after image of these different applications of visualization. But I want to come back to my 6 points, and I want to convince you about this whole thing with relationships and talk about the key things that really I think are driving visualization today. So the first one, size matters, right? So size matters, and we've seen examples of that already in visualizations today. A large visualization will slow us down. It'll remove our interactivity. A large visualization is something our computers can't deal with effectively. We have to find ways around that, but we have to be able to define the terms. And large is a term that is changing all the time. Large is becoming a larger term, a larger number, a bigger number. And so it's something that doesn't fit, right? Large is what we think is too big to deal with. So large can be something that doesn't fit into the GPU memory on our fancy graphics cards, and that's not very big at this point, 4 gigabyte maybe. Maybe it doesn't fit into main memory, so now 64 gigabyte is too large, gigabyte is too large. Maybe it doesn't fit on our local hard drives anywhere, so 10 terabytes is somehow too large. And so large is a relative term, and we have to deal with large in visualization all the time because the data sets are getting large. Here's an example of a 12 gigabyte data set rendered. This is from a micro CT data set. Here's an example out of a 600 gigabyte data set. This is obviously a landscape. This happens to be a big open pit copper mine that's in Salt Lake City that we see out our windows every day. And large can be coming from all different aspects of science and medicine. Large can come from small things. This simple, honest, or innocent-looking diagram here looks like a microscope image of some sort. Who knows what it is? It turns out that it's a single slice from a multi-section slice. There are 341 sections, 90 nanometer thick sections. Each one of these sections has 2 nanometer per pixel resolution. So this is a very high resolution image that was actually constructed by mosaicing, taking multiple scans in a microscope at the highest possible resolution and bringing those all together so that each one of the tiles, and this little block here is a tile, each one of those tiles is already 4,000 by 4,000 pixels. And there are about 1,000 tiles in each one of the sections you see here, and there are 341 of those sections. So we have a 32 gigabyte per section data set, and the whole data set, if you stack it up, is 16.5 terabytes. This is real data that we have to deal with from a collaborator who works in ophthalmology. These are sections to the optic nerve. This is real, relevant data. He wants to know connectivity of neurons through the optic nerve. He wants to create that functional link of information transfer through the optic nerve. He needs to analyze, he and his group need to analyze these sorts of data. So this is big as we deal with it. And we have tools to deal with this. One of the easy tools is to display this data over a large panel, to put huge displays together. So that the display of the resolution of the display starts to map the resolution of the data. That's one way to go about it. Another approach we're taking is embodied in a tool we call ImageViz 3D. And the idea of ImageViz 3D is that it handles a number of different data sets, has a number of different rendering tools built in, and it's very cross-platform. So that's sort of the computer science side of it. Inside, it has a rendering hierarchy. It has the image data, or the three-dimensional data sets, could be from images, could be from other forms of analysis, rendered at different hierarchies and different resolutions, and allow us to transfer and move through those different resolutions as the need arises, as the visualization and hardware support is available. And so we can produce images like this that show very high resolution pictures here of rat anatomy, it turns out, and then these human images here of the hand and the head. And I'll show you other examples of ImageViz 3D as we go along. Now, another thing I said is that portability matters. We're all sitting here with our laptops. I hope none of you are checking your email right now. But we're all used to carrying our computers around with us. We have our iPhones in our pockets. And so portable computing has made a huge jump, a huge transformation. And that's making its way into the world of biology and definitely into the clinical world. I deal with physicians now who think nothing of carrying their iPad around with them. They'll show their patients their images, the data they get from tests via an iPad. They're all dying for iPad apps for their particular discipline in medicine. And so portable visualization is our challenge. How do we make our visualization tools work in this range of computing scales and sizes and portabilities? ImageViz 3D is one of those tools. So this is the same image data set on a Powerwall and on an iPhone. ImageViz 3D can do this by having this hierarchical representation of the data, having it available in lots of different resolutions, and allowing the hardware to dictate and the patience of the viewer and the size of the data set to dictate which level of that hierarchy actually makes it to the screen. And so ImageViz 3D Mobile is an application you can download from the web right now. You can go to the iTunes store, and it has real-world capabilities that in the initial version, let's say, we're more toy. This is actually an iPhone 3, an iPhone 4. Obviously, it works differently. But this tool has found real application. This comes from a collaborative project we have at the University of Wisconsin with an investigator by the name of Chris Butson. Chris is a former student of ours, and he is actually working with physicians to render individual patient data in patients who are receiving deep brain stimulation. So these are electrical devices placed in specific regions of the brain to help calm motor disorders in patients. And so the placement of the electrodes is obviously critical, and the imaging tools that help direct that process are obviously critical. And so we can use ImageViz 3D so that physicians can actually see their patient-specific models, show them to the patient, work with the patient, and explain what's going on, and then use higher resolution tools, obviously, to do the rendering. So iPhone was a very cool device when it came out. It was a wonderful toy to show our applications on. As I've said before, the iPad or devices like it, tablet-sized devices, are I think going to have even a bigger impact in the clinical domain. They have, or they might have, the right sweet spot between resolution, computing power, and portability that make them a very attractive tool for physicians. And this is a platform we have to accept and learn to work with in our visualization software development. So I said before that perspective is important. And in that context in biology, it's always very important to know what you're looking at and what scale you're looking at, because biology happens across multiple scales. I'm here talking to you about high-scale things. You've seen wonderful images of brains. This is a set of data from Mark Ellisman at UCSD. This shows, sort of badly rendered here, the outline of the whole brain. Here's a piece of the brain, and you can zoom in on that piece of the brain, and you can zoom in even further on the structural elements of that brain. You can zoom in even further and get down to the cellular level and start to really understand connectivity. And that's how biology and that's how medicine happen. And so we have to have tools that can provide different resolution and deal with the fact that some things happen at resolutions that maybe are would say beyond what we can typically render, but the acquisition tools are now making those available to us. Here's another example of some data from Jacqueline Chen from Sandia National Labs. And these are the same images, left and right, shown at different resolutions. And Jackie gave a talk at SKI and contended, and she has other examples to show this, that the fine detail that you see in this high-resolution picture simply disappears in this low-resolution picture. And there are lots of examples we can find in science where resolution does matter, and it's critical to have the tools to support the resolution that's now becoming available on the acquisition. So many of us have worked with the digital human data set, right, maintained by the National Library of Medicine. Until recently, nobody knew that the visual man had tattoos. This, of course, is a breakthrough in science. This was front page New England Journal material, but it was made possible because we had rendering tools. This is rendering with ImageViz 3D again. We had rendering tools that could finally handle the full resolution data set that is available from the photographs taken as the visible man was sliced and created this data set. And so these actual pixels of ink are visible at the resolution that's available. But up until last year, it was not possible to render this much data at once. So there is data available that we're not taking advantage of. The other point I'd like to make, and I think we're all convinced of this, we've seen examples of this morning already, is that life moves, interactivity matters. We have to be able to deal with our data in an interactive manner. I'll show you this example in a test or a case study I'll talk about a little bit later on that will explain sort of what this all is, but it shows you moving around a three-dimensional torso model and interacting with that model. And that's the type of interactivity we all want in our data. And we have other examples of that. This is another piece of software called Map3D that we created many years ago and just today released the latest version of. Version 7 is now on our website. And the purpose of this tool was to render surface-based time signals. They arise in biology and in medicine all the time. You can picture that as the EEG signal on the surface of the scalp, or in this case, the electrograms on the surface of the heart, or the ECG on the surface of the body. And Map3D allows us to have captured the time signal aspects of it, but also the spatial distribution aspect of it. And this little animation here shows you a heartbeat. This is a heartbeat rendered in time as voltage mapped to color. This tool actually allows us to integrate a background image. This is an open chest of an animal. This is an animal experiment. We have an electrode sock around the heart. We're able to measure the voltages and project and render those voltages interactively in whatever time scale we want. We can grab this white bar here interactively, move it back and forth. We can change scaling options. It's got all this sort of quantitative analysis tools one could wish for for a guy like me in my sort of scientific geek world. We also can visualize clinical data this way. This is a picture of the left ventricle of a human. This is clinical data acquired with a semi-invasive catheter-based system. Map3D allows us to project that data onto the surface of the endocardium to look now at a high-scale view of activation of the heart as it circles around the chamber. So what we're seeing here is voltage evolving as a function of time. We're seeing a long run here. Each one of these peaks here corresponds to a heartbeat. We're seeing actually the same data set left and right here, but offset in time. So I'm able to compare the morphology of this beat with the morphology of this beat and see how similar they are. And that's a question that the clinicians had. And so we look for these tools that can interactively display our data. This could also move around in space. We can apply clipping planes, do all those things. This is easy in the sense that it's a small data set. It's surface bound. The resolution is not tremendous. And so this is easy to do in interactive terms, but lots of data sets are not getting this kind of treatment that really could use it. The other thing we heard in the previous talk actually came up as one of the questions was, what do we do in the area of uncertainty and errors? How do we deal with the fact that all our data have errors attached to them when it comes to visualizing those data? Right? Every good scientist knows they have a sense of how accurate their measurement tools are in the lab, and they can even tell us that. But we're very poor so far at incorporating that information in the visualizations that we give them back, or the tools we give them to visualize their own data. And as you can see, when it comes time to those critical areas where there's a lot of uncertainty and the decisions become important, that's when the stress level goes up, that's when the needs go up. And that, of course, is where clinical medicine comes in. I mean, what could be more stressful than making decisions about where you're going to cut somebody's brain based on their DTI images, let's say. And this happens. This is how clinicians plan their surgeries. They use the image data and they use the renderings and the visualization tools we give them to make real decisions about real people, maybe even one of us. And so this is a critical area of uncertainty. And Chris Johnson, the head of the Ski Institute, has sort of taken this on as his big project right now. And he likes to say, when is the last time you've seen an error bar in a three-dimensional visualization? Okay? And it's because they really don't exist. And here's an example of a brain and a tumor inside a brain. And that tumor gets segmented and forms this green blob here. And if you look closely at these borders here, somehow they get drawn. This one you can say, well, I can see where that border is. This border is somewhere in that gray mess. And yet we have to make a decision. We have to segment those data because we have to know where the tumor is. You have to tell the physician, where is the tumor? Where should the surgery be directed? And so we're trying to figure out new ways to capture the confidence interval around that border, to characterize the image content in a way that tells us something, at least, about the confidence we can have in that border and that segmentation. And so here's just a very simple example where we have these red and blue lines. The red line is the best guess, and the blue line is some notion of the uncertainty around that best guess. And we have to find ways to capture that information. This is a huge and challenging problem. It's a very non-trivial problem, and we're going to have to come up with very clever ways to encode that information in our renderings, in our visualizations, so that physicians and scientists can actually make use of it. It's an enormous problem. So, we're trying to validate our visualization. We're trying to use photographs to validate what we get from our image data sets and our rendering of those data sets. We're trying to do discrete measurements to capture the measurements we can make out of image data. And we're trying now to, again, find new ways to show the confidence values in the renderings themselves and somehow turn that into color in such a way that the physician at least has a chance to know how confident we are in the images we present. Now, of course, we all know that newer is better, right? We all update our computers regularly and can't wait to buy the latest iPad 2. And so newer is always better, and we're seeing technology come along. In the world of graphics and rendering and visualization, we've heard it a couple of times today already, GPUs are the term of choice right now. These are the graphical processors that we've enjoyed having in our computers to make our renderings more rapid. And now there are whole computer systems based on GPUs. There are GPU-based parallel architecture supercomputers that exist. And we're lucky we happen to have one of them. NVIDIA stole 2 of our best investigators away, and so in return, they gave us a lovely NVIDIA cluster. And so now we're a center of excellence for NVIDIA at Utah, and we have a box like this that allows us to start to play in this game and to use GPUs not just for visualization, although that's one of the application domains, but to use them even as a general purpose computing tool, because GPUs are very, very efficient tools. So our software has to take advantage of this, or take advantage of whatever is new that comes along. That places a huge burden on software engineering, and if you make use of the latest, greatest things, the minute you move to another platform, those latest, greatest things have changed. And so there's a huge challenge here, but we can't ignore this type of acceleration anymore. And GPUs have the potential to give us that. These are not trivial improvements. These are 10-fold, 100-fold improvements in certain algorithms. And this is what new means, I think, in the world of visualization, in the world of rendering. So now, in the last little part of my talk, I want to bring you a couple of case studies. The question came up of the utility of some of these tools to physicians. And I want to show you that in some cases, we're able to tie those tools together and actually deliver something that physicians are very keen to use and are very happy to deal with. And the first of those studies actually is based in Boston. I thought I'd pick a local one. This guy here is John Triedman, and he is a pediatric electrophysiologist. So he treats kids, and he puts defibrillators in kids. That's his colleague, Frank Chechen. This is our famous Matt Jolly, and Matt doesn't stay still for very long. So the only picture we could get of him was out kiteboarding one day. And these are a couple of folks at Utah who really developed all the technology and the visualization and the simulation results I want to show you. So ICDs are implantable cardiac defibrillators. These are devices that we have going into patients at over 100,000 a year. Now those 90,000 is an old number. I think the number is about 105,000 per year of these devices going into people in the US. So these are hearts that are at risk, hearts that have the potential to go into fibrillation, and when your ventricles fibrillate, you die. So it's a major health problem. However, these devices are also finding their way into children. This is where pediatric cardiologists come into play here. And so in children, this device, which looks so tiny in the hand of a human here, suddenly becomes a pretty big piece of hardware to put into a child. You're also putting a piece of hardware into a child that's going to stay there, presumably, for most of the rest of the life of that child. That risk of fibrillation does not go away. We don't cure this very well. This is one of the holes in medicine right now. And so having that device in a child brings in a whole different set of problems. And so the resulting techniques physicians use span quite a spectrum. So here's a placement of the device under the left clavicle. This is kind of the traditional placement of the device. This is what we do in adults. Straightforward, no problems. You see the wires coming down here. The wires go into the heart. One of the wires, I don't know where this one goes, disappears somewhere quite low. But somewhere in here is the heart of the child, and these electrodes will then be used to shock the heart back into normal rhythm. Here's another case. The device, you can see, is enormous compared to the thorax of this child. You can see the rib cage here. So that device was too big to be placed under the left clavicle. That device got placed in the abdomen of the child, and the electrodes, you can see, are in totally different locations. Here's another case where the device ended up in the pelvis of the child, and again, the electrodes are in a range of locations, and there's a range of numbers of electrodes. First case has 2 electrodes. This case has 1, 2, 3, 4, at least 4 electrodes, and the case itself is a potential 5th electrode. So there's a lot of variation here. The physicians have no guidance in developing those placement strategies. They have no tools with which to do that. And so we developed a pipeline based on using images to create patient-specific models, solving some simulation problems, predicting the electric field on the heart from the device, given a certain electrode location and a shock protocol. And then we're able to deliver that to the physicians to allow them to help make their decisions. And so there's lots of very cool rendering of geometries here. Here you see the torso model. Here's the device itself blown up. Here's some renderings, again, of the model, the structure of the mesh we use, and some results presented on there. And I want to show you now this visualization, this animation I showed you in the beginning. This is what the physicians see when they actually place the device. So they have a three-dimensional model of the thorax that we've created for their particular patient. They can go in and grab these things over here, which are the catheters, the electrodes, and they can place those electrodes. Here's an example here where the physician's moving that electrode around, deciding where it should go in the heart, and then is able to save that placement as a case, run the simulation to predict what type of shock would be created, and then visualize the result of that simulation. So again, interactive visualization on a fairly large scale. These are thoracic models. These are not small. These are based on MRI and CT data. These are interactive slices now through the thorax. This is all very much in real time. It's a direct screen capture. This is showing the voltage gradient. We can then predict the actual current density. That's the quantity of interest electrophysiologically. And again, there's this three-dimensional interaction of time and space that's possible with these tools. We can do some simple visualizations, too, for the physicians, in which we say, well, here's the histogram of the size of the voltage gradient for all those voxels that belong in the heart. And here's those that are below a threshold of 5 volt per centimeter, and here are those that are above another threshold of 30 volt per centimeter. And it's the green area here that's of interest. We want to have the maximum amount of the heart in that nice sweet spot of between 5 and 30 volt per centimeters. That's the most effective defibrillation shock. And we can, again, present that to the physicians. We can give them a display like this that shows all aspects of the data. And this they're using. So we're validating this now in our studies here in Boston and in Utah with actual pediatric cases of ICDs. We've got to the point now where we were doing validation studies. That means we were just recording and using their measurements to support our simulation data. Now we're to the point where the physicians, if they have a case that's particularly troublesome, they're coming to us and saying, I know you're doing just validation, but could you make the model for me? Could you let me put some different configurations together? Because I really am not sure with this kid where I want to put the device. And that's the place we've been able to achieve with a tool that really meets a demand for them. Another example, totally different field, similar type of situation. This is the world of osseointegration. This is an amputee, and you see here that he has not a stump and a socket. He has an attachment to his residual limb. This is what the attachment looks like. This is a dramatic transformation in the world of prosthetic devices for these often young and healthy, often wartime veterans who need orthotics, need prosthetics and are looking for a new way to get it. We developed, again, a pipeline based on patient-based, subject-specific modeling, image-based models that we created, and then we're able to visualize the electric field around this device when we implant it, because it turns out if you apply electric fields to bones, they grow better. And so we came up with a configuration where you can apply an electric field to this implant, which is a metallic rod, and then have bands around the limb that describe the other pole of the stimulator, and then predict what the electric field will look like in that region of ingrowth. And again, this is a tool that's going off now into some clinical trials, now into some device development, and this Joe video was an editor's choice, actually, based on this. So the last case I want to talk about, the last sort of case study, is visualization atrial fibrillation. So atrial fibrillation is another disease that affects the heart, and this time it's the atria that contract out of synchrony. You don't die from this immediately, but your chances are much worse. So there's a high risk of death by stroke eventually, and injury by stroke in the case of AFib. And this is a group that I work with now, helped found a few years ago, called CARMA. It's the Comprehensive Arrhythmia Research and Management Group. And it's a group very interdisciplinary of computer science types over here. Ross Whitaker, some of you may recognize, physicians and imaging acquisition specialists here, and then lots of great staff to make things happen. This is a visualization. I hope you can see it well enough. This is a nice, simple example of a visualization. This is a cine MRI. We've got some great background in MRI, so you know what this means. So here we have normal contraction. Here we have atrial contraction there, followed by ventricular contraction. Atrial contraction, ventricular contraction. Over here we have atrial flapping about. uncoordinated atrial mechanical activity. This is what AFib is. This is another use of visualization, very simple. This is one of the most effective pictures that we use in our group to explain to people what AFib looks like. It doesn't have to be fancy, color, or complex to convey a message. Now, there's a number of aspects of visualization that come into looking at the raw data. You've seen some great examples of that in the world of brain MRI. We do the same thing in the heart. But we take it a step further. We take our images here, which are maximum intensity projections here of images of patients who have AFib and are treated in a certain magic way in the MRI scanner, and we use simple things like color rendering. We simply do a color lookup rendering here to highlight these enhanced areas here with some green and yellow colors. We just apply a simple color map, and already the physicians are thrilled. This is so much more visible to them than this. Now they're used to seeing the green and the yellow, and it turns out green and yellow is bad. And sometimes that's the way physicians like to think about things. The more green, the more yellow, the worse off these patients are. And the worse off their chances are of successful cure using the techniques that we use. And so we've developed a bunch of tools for processing image data for this. These tools go into the clinic every day. This is CoreView. This is the tool that combines rendering of those MRI images where we can slice through the whole heart with segmentation of those regions of enhancement here, those bright regions, combined with three-dimensional rendering of the left atrium showing the green and the blue and the colors that the physicians like to see and like to use to evaluate these particular cases. Ablation is the cure method or the method of treating these patients. Nobody likes to use the cure word yet, but it's a new treatment that involves putting catheters inside the heart, going into the left atrium, and applying radiofrequency energy to disrupt this abnormal, uncontrolled electrical activity. And here again, visualization plays a key role because we're developing tools that allow us to use real-time MRI to guide the placement of the catheters, to guide the visualization of the lesions that these catheters here that are delivering the radiofrequency energy will actually provide. And so we get animations like this, in which you see in real time the MRI image updating with a rate of a couple of frames per second. You see the catheter here is a cartoon superimposed on that. And then in green, if you look carefully here, you actually can see the outline of the right atrium in this case. This is an animal study. And so here in this visualization, we're bringing real-time visualization of the images combined with previously acquired segmentations of that particular anatomy, combined with animation of this catheter based on location information of coils embedded in that catheter, and we're bringing all that together into a real-time visualization environment that the physician has on a screen as they work on the patient. And so this is visualization making its way into the actual clinical procedure room. This is the real thing, I think, in terms of that ski vision I showed you in the beginning, of taking visualization, image analysis, and tying it all together, and we're actually beginning to do it. So, as I said, I think there's a lot to be said about the relationship between visualization and relationships. A lot of the same factors matter. If this helps you remember some of those factors, then it's a good analogy, a good metaphor. I have to, and happily will give lots of credit to the wonderful people at the Ski Institute who have generated most of the software and the visualizations you've seen. And I would encourage you to come visit us and come visit our download site, because many of the tools you've seen on display here today are available right now on the website and are freely available for your download and use. And we do have seminars and workshops that we hold, one of them coming up in the next couple of months, to help teach people how to use these tools in this domain. And so with that brief excursion in the world of sort of function and physiology and medicine, I thank you for your attention very much.